Thank you for the invitation. Let me start my presentation.
Research work done in my laboratory using the CRISPR-Cas9 technology. Today's presentation will be divided in two parts. I will first present the use of the CRISPR-Cas9 technology to treat hereditary disease, and my graduate student, Gabriel Lamothe, will present how the CRISPR-Cas9 derived technology may be used to detect viral infection. The word CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. CRISPR-Cas9 technology is derived from research on bacteria. It was initially discovered in 2005 that the CRISPR sequence contained viral sequence, and yet there was an hypothesis that CRISPR-Cas9 is the bacterial immune system. It's only in 2013 that it was discovered that the CRISPR-Cas9 may be used to induce specific modification of the human genome, and this started a big explosion of articles. Indeed, the CRISPR-Cas9 system is used by bacteria to kill bacteriophage. Bacteriophage are extremely abundant. They're 10 times more abundant than bacteria, and there is a war that is ongoing between bacteria and bacteriophage since millions of years. It was initially discovered that there are in the genome of bacteria constant repeated sequence. Researchers were wondering what these repeated sequences were for, and then they discovered that the sequence between the constant repeat were sequences that were from the genome of various bacteriophage. In fact, what normally happens is when a bacteriophage infects the bacteria, normally the bacteriophage wins, the bacteria is dead. But sometimes you have a defective virus that will infect without killing the bacteria. Bacteria will then be able to acquire some part of the sequence of the virus, and it will store that into its genome, and it will be ready when it needs such bacteriophage again in the future. From the DNA in the CRISPR RNA, bacteria will first express a pre-CR RNA, and then there will be a complex that will be made between tracker RNA, CR RNA, and the Cas9 protein. This tracker RNA, CR RNA, Cas9 protein complex will then bind with the bacteriophage DNA and induce a cut of the bacteriophage DNA to essentially kill the virus. To bind to the DNA, the Cas9 protein requires the presence of a protospacer adjacent motif, which is simply NJJ for the Streptococcus pyogenes tab. Then there is the CRRNA, which contains a variable sequence of 20 nucleotides that will be complementary to the bacteriophage sequence. And then there is also the tracker RNA, which is a constant RNA sequence that forms a complex with the CRRNA. When the three parts are together, the CRRNA, tracker RNA, and the Cas9 protein, a cut will be induced at exactly 3 nucleotides from the plant. Of course, since this is a war, there are countermeasures that are taken by the phage. They will mutate their genome so that they become resistant. They are not recognized by the CRRNA and will survive bacterial defense. The CRISPR-Cas9 Big Bang started in 2012 when Genex and colleagues realized that the CRISPR-Cas9 technology can permit to cut the genome of plants and animals. They also fuse the CRRNA and the tracker RNA together to form a single guide RNA. This was done essentially because the single guide RNA is something that can be patented, whereas the CRRNA and the tracker RNA, being natural, they cannot be patented. During the following year, there was an explosion of articles confirming that the CRISPR-Cas9 technology may be used to cut the genome of bacteria, human cells, farm animal, mouse, frog, zebrafish, plant, flies, nematode, yeast, monkeys. Many of these articles were published in prestigious journals such as Nature, Science, Nature Biotechnology, Molecular Cell, Molecular Cell. There was a rapid explosion of articles, and as you can see, as of November 26, 2020, 21,420 articles have been published where the word CRISPR is mentioned. The main reason of this rapid explosion of articles is that it costs much less to derive a sequence that will be able to cut a specific genome sequence compared to other pre-existing technology such as zinc finger protein and TALENs. As mentioned before, the binding of the Cas9 protein to the DNA requires the presence of a photospacer adjacent unsafe PAM, which is NJJ for the Cas9 derived from Streptococcus pyogenes. There is then the binding also of the single guide RNA, which will recognize the sequence of 20 nucleotides. And when the complex is made between the Cas9 protein, the single guide RNA, and the DNA, there is a cut that will be made at exactly 3 nucleotides from the PAM.
Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version.
One of the reasons of the rapid explosion is that the CRISPR-Cas9 technology also used all what was developed by gene therapy in the 30 years that preceded it. We have methods of delivering the DNA using AAV, adeno-associated virus, cationic polymers, liposome, microinjection, electroporation. All of those techniques are used in the context of CRISPR. When a double-strand break is produced in the DNA, most of the breaks will be repaired by non-homologous conjoining that will lead to micro-deletion or micro-insertion. And this is frequently used to knock out the expression of a gene by changing the reading frame. And it can also be repaired by homology-directed repair, which require the presence of a donor DNA that contains sequence of homology with what is preceding, what is following the cut, and in between the two homology sequence, there is a sequence in blue, which may be a single nucleotide or a whole gene that will be inserted at the site of the double-strand break. When we use the CRISPR-Cas9 technology, we intend to produce a double-strand break at a precise site in the human genome. However, the Cas9 may sometimes induce cut at other sites in the genome, which are called off-target mutation. Computer software may predict these off-target mutations by using the whole human genome sequence. However, these softwares are not perfect, and they will sometimes predict off-target mutation at site where there is no cut, and sometimes there will be a cut at a site which is not predicted by the computer. There are, however, some experimental techniques, such as the guide-seq method, which permit to experimentally identify the sites of off-target mutation. This guide-sec technique used the introduction of a short 34 base pair oligonucleotide at the site of the cuts, and later by PCR, the site of insertion may be identified by sequencing. The presence of these off-target mutations is the main problem that delays the use of CRISPR-Cas9 technology for direct in vivo corrections. One of the methods to reduce off-target mutation is simply to reduce by 2 or 3 the number of nucleotides of the single guide RNA which is binding with the target DNA. Another method is to use a mutated Cas9 nuclease that cut only one strand of DNA. It is then called a nickase. There is variation of Cas9 where the nickase cut only the lower strand of DNA, and other mutation of Cas9 which permit to cut only the upper strand of DNA. It is thus possible to induce more precise double-strand break in the DNA by using the Cas9 nickase and 2 single guide RNA, each detecting a sequence of 20 nucleotides close to one another. It is also possible to use a non-functional Cas9, a dead Cas9 nuclease, fused with the FOC1 nuclease. Then 2 guide RNA are again needed to detect sequences which are close to another, and the FOC1 nuclease need to form a dimer in order to be able to cut the DNA. Finally, some researchers have mutated the gene coding for Cas9. They have modified some sequence coding for amino acid to reduce the non-specific binding between the Cas9 and the DNA. The SpCas9 gene is too big to be inserted with a single guide RNA inside an adeno-associated virus, and thus researchers have identified Cas9 from other bacteria that are smaller. For example, the Staphylococcus aureus Cas9 is smaller than the SpCas9, but it has a different path which is more restrictive. Another type of CRISPR-Cas9 enzyme has been identified. It's called CPF1. It produces sticky answers in the blunt cut of the DNA. There is thus a whole series of various Cas9 obtained from various bacteria that require different paths. The CRISPR-Cas9 technology may be used not only to induce cuts in the DNA, but it may also be used to induce the expression of a gene by fusing Cas9 with BP64, for example, or fusing Cas9 with CRAB to repress the expression of a gene. In my research group, we are using the CRISPR-Cas9 technology to develop therapies for various diseases, Friedreich ataxia, Duchenne muscular dystrophy, and Alzheimer's disease. I will present that now. We have initially used the CRISPR-Cas9 technology to develop a treatment for Friedreich ataxia. It is an hereditary disease due to the presence of a long trinucleotide repeat, GAA, in intron 1 of the frataxin gene. This long repeat reduces the expression of the frataxin gene, leading to neurological problems and cardiac problems. We have thus used the CRISPR-Cas9 technology and generated single guide RNA able to cut in intron 1 before and after the trinucleotide repeat, leading to its removal and increasing the expression of frataxin. Indeed, the removal of the trinucleotide repeat in cells of the YG8-SR mouse model of Friedreich ataxia doubled the expression of frataxin compared to the untreated cells and raised the expression of frataxin to almost the normal level. The SPCas9 gene is too big to be delivered with 2 single guide RNA by a single AAV. We are thus currently using the smaller CGCas9 gene to remove the trinucleotide repeat in intron 1 of the frataxin gene. Using this smaller CGCas9 nuclease, we are also able to remove the trinucleotide repeat in intron 1 of the frataxin gene. We are also using the CRISPR-Cas9 technology to develop a treatment for Duchenne muscular dystrophy. Duchenne muscular dystrophy is due to mutation in a gene coding for the dystrophin protein. It is a large gene containing 79 exons. Some of these exons do not contain a multiple of 3 nucleotides, and thus 70% of Duchenne muscular dystrophy cases are due to deletion of one or several exons, and the total number of coding nucleotides which are deleted is not a multiple of 3. For example, in this presentation, we have a deletion of exon 50 that does not contain a multiple of 3 nucleotides. This leads to a frame shift, and there will be a stop codon in exon 51. Thus, the beginning of the dystrophin protein is expressed, but not the end of the dystrophin protein. This situation may be corrected by inducing the skipping, that is the removal, of exon 51 in the messenger RNA. This is done using antisense oligonucleotide. This exon 51 may also be deleted by inducing cuts in intron 50 and in intron 51 to completely remove that exon. This results in the expression of the beginning of the dystrophin protein and of the end of the dystrophin protein, but there is, however, a small part of the protein which is missing in the center of the protein. The removal of one or several complete exons may thus restore the reading frame and convert a Duchenne patient into a Becker patient. However, some Becker patients have severe symptoms and are bound to a wheelchair at the age of 11. Therefore, the improvement of some Duchenne muscular dystrophy may not be significant. This is because the dystrophin protein has a complex structure. Indeed, in its central part, the dystrophin protein contains 24 spectrin-like repeats. Each spectrin-like repeat is made of 3 alpha helix, helix A, helix B, and helix C. Note that helix A is starting on the left side and helix C is ending on the right side. And normally there is a succession of ABC, ABC, ABC. The main problem of deleting complete exon to restore the expression of dystrophin protein is that the beginning and the end of the spectrin-like repeat, indicated in this scheme by the black arrow, do not correspond with the beginning and the end of exons. And thus, when removing complete exons, the resulting of the spectrin-like repeat structure may not be normal. Becker muscular dystrophy patients have a deletion of one or several exons, but the total number of coding nucleotides which are deleted is a multiple of 3 nucleotides, and thus there is no frame shift. This is the case, for example, for Becker patient having a deletion of exon 45 to 47 or having a deletion of 45 to 49. In both cases, there is no frame shift. The beginning and the end of the dystrophin protein are expressed. However, at the junction site between the remaining codons, there is an abnormal structure of the dystrophin protein, and due to this abnormal structure of the dystrophin protein, these Becker patients are bound to a wheelchair at an early age. Thus, in my research group, instead of trying to restore the normal reading frame by deleting complete exons, we have instead aimed at producing an hybrid exon 5054, which not only restores the normal reading frame, but codes for a dystrophin protein with a normal structure. We have done our initial experiments using the myoblast of a Duchenne patient having a deletion of exon 51, 52, and 53. Thus, because the number of coding nucleotide was not a multiple of 3, there was a stop codon in exon 54 due to the frame shift. We have thus used the CRISPR-Cas9 technology to induce a cut in exon 50 and a cut in exon 54 to create the hybrid exon 5054. To produce this hybrid exon, we initially identify what are the possible SP-Cas9 PAM, or what are the sites where we can cut in exon 50 and in exon 54. We identify 10 PAM sites in exon 50 and 14 different PAM sites in exon 54. This table predicts what are the results of inducing cuts in exon 50 and in exon 54. When we have a blue square, it means that the cut has been produced in exon 50 right after the 3 nucleotide that codes for an amino acid. And the cut has been produced in exon 54 just before the 3 nucleotide that codes for an amino acid. Thus, at the junction point, we have amino acids that are, one is coded by exon 54, followed immediately by an amino acid coded by exon 54. There's no frame shift and there is no new amino acid produced. However, when the square is green, that means that we have at the junction site a new amino acid which is produced because we have cut in exon 50 right after one of the nucleotide of the codon. And the 2 nucleotides that will complete these codons are from exon 54. We have cut right after one nucleotide of the codon in exon 54, and thus there's 2 nucleotides left to create a new codon at the junction site. There's thus a new amino acid at the junction site, but there is no frame shift, and all the other amino acids are the correct ones. It is also possible with the square in red to have a new codon produced at the junction site, but this new codon is a stop codon. This is not what we want to do. All the squares in white are because there is a frame shift. We're cutting after one nucleotide of a codon in exon 50, and we're cutting before the last nucleotide of a codon in exon 54, and thus at the junction site, there is a new codon which is done because there is a frame shift, and all the amino acid that follows are not the correct one. We have thus tested various pairs of guide RNA. One guide RNA cutting in exon 50, and the other guide RNA cutting in exon 54. This produced an hybrid exon 5054 that had the predicted size of this hybrid exon. Note that when we are doing cuts in exon 50 and in exon 54 in a normal dystrophin gene, we are deleting 160,000 base pair. And despite that, the junction between exon 50 and 54 is of the size predicted. We have sequenced this hybrid exon that was produced by cutting in exon 50 and in exon 54. Not only were the hybrid exon exactly the size as predicted, but the sequence were also exactly as predicted. For example, we had at the junction site the fusions of exon 50 and exon 54 producing exactly the predicted amino acid. We also obtained at the junction site a new codon coding for the predicted amino acid or coding for a stop codon, and sometimes, as predicted by the white square, we had a frame shift, and when there was a frame shift, there were stop codons that were met in the resulting hybrid exons. As mentioned before, the dystrophin protein contains in its central part 24 spectrin-like repeats, each one being made of 3 alpha helix A, B, and C. We have thus used a guide RNA to induce a cut in exon 50 in a sequence coding for helix C, and another guide RNA inducing a cut in exon 54, also in a sequence coding for helix C. The resulting hybrid exon 5054 codes for an hybrid helix C, where the beginning of helix C is coded by exon 50 and the end of helix C is coded by exon 54. This is a structure that has been computer predicted using the sequence of the resulting protein. When the myoblast of this Duchenne patient having a deletion of exon 51 to 53 are fused together to form some small muscle fibers in culture called myotubes. In fact, these muscle fibers do not express dystrophin because this is a Duchenne patient. However, the myoblast of a healthy human, when they fuse together, they form a small myotube and they do express dystrophin, as can be seen here in this Western plot. When we use the myoblast of the Duchenne patient and we induce the formation of the hybrid exon 5054, we have expression of the dystrophin protein in culture. And as you can see, this dystrophin protein has a lower molecular weight than the normal dystrophin because there is deletion of exon 51 to 53 and an additional deletion of part of exon 50 and part of exon 54. Thus, there is a part of the dystrophin protein which is absent, and this is why the protein is smaller, but it is nevertheless expressed. For our initial in vivo experiment, we have used the HTMD mouse model. This is a transgenic mouse that expresses the human dystrophin gene, all introns and all exons. We have electroporated in the muscle of this mouse the plasmid coding for the SPCas9 fused with the fluorescent green protein and 2 single guide RNAs. This a month later, when we took the muscle, there was expression of the green fluorescent protein, confirming that the plasmid had been correctly electroporated in the muscle fibers, leading to the expression of the fluorescent green protein, and probably also leading to the expression of the SPCas9 and of the 2 guide RNA. We then extracted the DNA from these muscle, and we first confirmed using a test called the surveyor enzyme that indeed there were cuts that had been produced in exon 50 and 54. We then used PCR to amplify the hybrid exon 50-54. Note that in the muscles that are not treated with the CRISPR-Cas9 technology, there is no amplification of exon 50-54 because there are 160,000 base pairs between these 2 exons, and therefore the 2 primer for the PCR are too much separated from one another. We then sequenced this hybrid exon 50-54 that was produced in vivo in the mouse muscle expressing the human dystrophin gene. And as predicted, we had the sequence of exon 50 and a new codon at the junction site, followed by all the correct codon coding for the normal amino acid in exon 54. This is exactly as predicted in vivo. However, for delivery of the Cas9 gene in vivo to several muscles, we need to use an adeno-associated virus. As mentioned before, the SPCas9 gene is too big to be delivered with 2 guide RNA by a single AAV. And we therefore use for our next experiments the Cas9 of the Staphylococcus aureus, which is a smaller Cas9, which permit delivery with 2 guide RNA by a single AAV. In this case, we have used guide RNA that induce cuts in exon 47 in a sequence coding for helix B, and in exon 58 in a sequence again coding for helix B. This resulted in the formation of an hybrid exon 4758 coding for an hybrid helix B, which has a normal structure. The beginning of the helix B being coded by exon 47 and the end of that hybrid helix B being coded by exon 58. We then use for our in vivo experiment a new mouse model called HDMD Delta 52. It's the same mouse model as previously that contained the dystrophin gene with all the exons and all the intron, except that this mouse has a deletion of exon 52, and thus there is no expression of the human dystrophin gene. We have thus used the CRISPR-Cas9 technology to induce a cut in exon 47 and a cut in exon 58, producing the hybrid exon 4758, resulting in the production of a dystrophin protein containing an hybrid exon, hybrid helix B. We have injected to these HDMD Delta 52 mouse AAVs coding for SACas9 and 2 single guide RNA. A month later, we observed in the muscle the expression of the human dystrophin gene, including in the heart. We are thus proposing a treatment of Duchenne muscular dystrophy, which would be the systemic delivery by an adeno-associated virus of the Cas9 gene and of 2 guide RNA to form an hybrid exon. In contrast with exon skipping, which is a treatment done at the level of the messenger RNA, the treatment that we propose at the level of the DNA would be permanent. The CRISPR-Cas9 technology is evolving rapidly, and new technologies derived from the CRISPR-Cas9 permits now the modification of a single nucleotide. More than 32,000 single nucleotide modifications are responsible for hereditary disease, and thus the capacity to correct a single nucleotide would provide treatment for most of these hereditary disease. The first treatment which permit the modification of a single nucleotide was developed by Kamar et al. In 2017. It used a Cas9 nickase, that is a modified Cas9, which is able to cut only one strand of DNA, and this Cas9 nickase is fused with a cytidine deaminase. This technology permits to chemically modify cytidine into a uridine, which is replaced by a thymine following DNA repair. The main limitation of that technique is the chemical modification of the cytidine will occur in a narrow window of 5 nucleotides located at 12 to 16 base pair from the NGG path. We have initially used base editing technology to develop a treatment for Alzheimer's disease. The Alzheimer's disease is produced by an abnormal metabolism of the amyloid precursor protein. Normally, this protein is cut by the alpha secretase, followed by a cut by the gamma secretase. This produces peptides and protein fragments that are degraded without causing any problem. However, this protein may also be cut by the beta secretase, followed by a cut by the gamma secretase, and this produces short 42 amino acid long beta amyloid peptides that aggregates to one another forming amyloid plaques that interfere with synaptic transmission, leading to neuron death and memory problems. This scheme illustrates the amino acid sequence of the transmembrane part of the amyloid precursor protein. We can see the position of the beta, alpha, and gamma synthetase cut site. All the amino acids with the star above their name are amino acids which are modified, leading to familial form Alzheimer's disease. Please note the position with the red arrow. This is the alanine in position 673. When this alanine is changed by valine, this leads to severe early onset Alzheimer's disease, and you are Alzheimer at the age of 40. However, when this alanine is changed by threonine, you're not Alzheimer, even when you're 95, 100, or 105 years old, as shown by Johnson et al. In Nature 2012. The presence of the A673T mutation, also known as the Icelandic mutation, reduces the secretion of A-beta 40 and A-beta 42 peptide for the wild-type APP gene and for APP gene containing the London mutation. Our experiments have shown that the presence of the A673T mutation reduced the secretion of A-beta 40 and A-beta 42 peptide by the APP genes, not only for the wild-type gene, but also for APP genes containing several familial Alzheimer's disease mutations. As mentioned before, the CRISPR-Cas9 base editing technology permits to modify the cytidine into a thymine. We thus have used the base editing technology to target the cytidine in the antisense strand of the alanine codon, transforming that cytidine into a thymine and just changing the alanine codon into a threonine. The main problem with this approach is that although we want to modify the cytidine into the antisense strand of the alanine codon, there are other cytidine nucleotides nearby that are also affected by the base editing approach. We have constructed 14 different base editing enzymes to be able to modify more specifically cytidine in the antisense codon of alanine. By modifying the cytidine in the antisense codon of alanine, we have been able to introduce the A673T mutation in up to 17% of the APP gene. However, other cytidine, also located in the antisense strand, were also modified by this approach. A new fantastic technology called prime editing has recently been developed by Anselon et al. It permits, in principle, to replace any nucleotide by any other nucleotide. The prime editing technology uses a Cas9 nickase fused with a reverse transcriptase. It also requires a prime editing guide RNA known as a PEG RNA. The PEG RNA is essentially a prolonged single guide RNA. Thus, as the single guide RNA, it contains a spacer sequence which react with 20 nucleotides in the target DNA. It also contains the constant scaffold of the single guide RNA, which is in red, and then at its 5' end, there is a prolongation with the primer binding site, which is a sequence of 10 to 17 nucleotides reacting with the upper strand of DNA. This is followed by the reverse transcriptase template again, which is 10 to 17 nucleotide in length, and which will contain some modified nucleotide, in red in this case, indicating which nucleotide has to be modified by the reverse transcriptase. A plasmid designed by Anzalone et al. Is available at AdGene to construct new PEG RNA. This plasmid contained the red fluorescent protein gene, which may be removed by BSA1 cuts. This produces a backbone of the protein, and then the other components are the spacer, the primer binding site, the reverse transcriptase template, and the PEG RNA scaffold. All of these sequences are single-stranded oligonucleotide that may be purchased from IDT. These four parts are then assembled together to produce a new PEG RNA. To use the prime editing technology, we first have to identify photospacer adjacent motif, PAM, which is NJJ for the Cas9 of Streptococcus pyogenes. This will permit to the Cas9 to bind to the DNA. Then the spacer sequence of the PEG RNA will bind to a 20 nucleotide sequence of DNA, in this case the lower strand, and the formation of the complex between the PEG RNA, the Cas9 nickase, and the DNA will induce a nick at exactly 3 nucleotides from the PAM in the upper strand of DNA. This will release the upper strand of DNA to be able to interact with the primer binding side of the PEG RNA. And then the reverse transcriptase template, which contains a few nucleotides to be mutated, will be available for the reverse transcriptase to synthesize a new DNA upper strand. Many Duchenne muscular dystrophy patients have a stop codon in the DMD gene. Since we did not have access to cells of patients containing such point mutation, we decided to introduce these stop codon using the prime editing technology. So in each case, we had to identify an NGG PAM, identify the PEG RNA photospacer sequence, and then modify the reverse transcriptase template so as to modify a codon for an amino acid into a stop codon. In this case, we have changed cytidine to thymine to introduce the TGA stop codon. We have successfully used that approach to introduce stop codon in exon 9, 20, 35, 43, 55, and 61. We have thus designed various PEG RNA targeting DMD exon 35. As you can see, we have varied the reverse transcriptase template in blue from 10 to 16 nucleotide, and we have also varied primer binding site in green from 10 to 15 nucleotide. And at the desired mutation site, we have introduced a T to introduce that mutation. Our initial experiments were done in HEK293 T cells. We tried to reproduce the mutation of MX1 gene and to target DMD exon 35. So essentially, these cells were transfected with plasmid coding for the Cas9 nickase fused with the reverse transcriptase and a PEG RNA targeting either exon 35 of the DMD gene or the EMX1 gene. The DNA was extracted 3 days later, and the targeted sequence was PCR amplified and sequenced using Sanger method. The sequence were analyzed using the EDIT-R online program. Essentially, we observed a 32% correction of mutation of the AMX1 gene as done by Enzalone et al. However, for the exon 35 mutation, we had a 2% background in the sequence of the control, negative control. And with the different PEG RNA, we have a mutation ranging from 4 to 8%. So this was not as great as for the EMX1 gene. We have thus tried different methods to try to increase the percentage of genome editing of exon 35. The first method that we have tried is to repeat the treatment 3 times. Essentially, the cells were transfected with plasmid at day 0, 6, 12. And DNA was extracted 3 days and 6 days after each treatment. We have amplified by PCR exon 35 at each of the extraction day and sequenced it by Sanger and analyzer sequence. As you can see, the percentage of genome editing increased from day 3 to day 18 with the repeated treatment. And this was the case for all 3 PEG RNA that were targeting exon 35. We have then tested a second method to try to increase the percentage of mutation in exon 35. It is to induce a second nick in the target gene. This is the PE3 method. Essentially, we have identified 2 PAM sequences, which permitted to guide RNA to induce a second nick at either 57 nucleotide from the original PEG RNA nick or at 24 nucleotide from the PEG RNA nick. When we induced a second nick at 24 nucleotide or at 57 nucleotide from the nick induced by the PEG RNA, there was a significant increase in the addition of the target gene for PEG RNA 35.4 and PEG RNA 35.6, but not for PEG RNA 35.5. We still have to understand why. A third method to improve the percentage of mutation in the target gene is to mutate the PAM used by the PEG RNA. We thus designed PEG RNA that were not only able to introduce a stop codon mutation, but were also able to mutate simultaneously the PAM that they use. Mutation of the PAM used by the PEG RNA improved the percentage of mutation in the stop codon for 2 of the 3 PEG RNA that we have tested. Combining the 2 methods, that is, inducing a second nick in the target gene and mutating the PAM used by the PEG RNA, further increased the mutation of DMD exon 35 to 39% with all 3 PEG that we have tested. We are currently starting new project to correct mutation responsible for other hereditary disease. We are working on cystic fibrosis due to mutation in the CFTR chloride channel, on congenital muscular dystrophy due to mutation in the ryanodine receptor, and on spastic ataxia 8 due to mutation in the NKX6 type 2 gene. For each hereditary disease due to a point mutation, for example here with spastic ataxia type 8, it is possible to correct in principle the mutated gene using the prime editing technology. In this case here, we can identify that the mutation is an adenosine change for thymine, and thus we can identify a PAM NJG for the SP-Cas9, which is close to the mutated nucleotide. We can then design a PEG RNA that will introduce the desired mutation. In this case, we are introducing two mutations. One to correct the mutation, to reverse the thymidine into an adenosine. And the second point mutation is to modify the PAM so that following the correction, the Cas9 enzyme can no longer bind to the DNA. CRISPR-Cas9 derived technologies may thus be used to treat many hereditary diseases. The main problem remained the in vivo delivery of the editing agents. Thank you for your attention.
Okay, thank you, Professor Jacques. I'll take the turn now to make a general comment for those who don't speak English. Okay, thank you for your presentation.
Thank you.
Of course. Boa noite a todos. Meu nome é Matheus. Eu trabalhei como professor de inglês durante uns 2 anos e assisti a palestra junto com vocês e vou tentar passar um apanhado geral do que o professor falou. É claro que em poucos minutos eu não consigo passar a ideia e mesmo se eu tivesse todo o tempo que ele teve para poder fazer essa apresentação, eu não conseguiria passar porque eu não tenho a mesma expertise dele. Então, ele começou falando sobre a tecnologia CRISPR-Cas9, assim mesmo se pronuncia. Eu me adianto e peço desculpas pelos meus erros de pronúncia com relação aos termos biológicos, porque eu não sou da área, não conheço muito bem, mas vou tentar ajudar vocês a entenderem de uma forma geral. Podem comentar aí no chat, caso vocês não estejam entendendo, e me pedir que eu possa repetir alguma coisa. Então, a tecnologia do CRISPR-Cas9 é a derivada de pesquisas com bactérias, como ele mostrou nos slides, e somente em 2013 é que foi descoberto que essa tecnologia pode ser usada para induzir modificações físicas no genoma humano, e não só humano, como de outros animais, também foi mostrado por ele. E isso gerou uma grande explosão de artigos e pesquisas na área. Por que gerou essa grande explosão? Porque ao mesmo tempo em que um RNA guia, que foi o termo que ele usou, um único RNA guia para fazer um processo, é algo que pode ser patenteado quando se usa um processo natural, com múltiplos trabalhos de RNA, ele usou o termo RNA-CR e RNA-trans-CR, são processos naturais e que não podem ser patenteados. Então, passa a custar muito menos derivar uma sequência que será capaz de cortar uma parte específica do genoma, seja humano, seja de outros animais, em comparação com outras tecnologias pré-existentes, como a da proteínas NF ou do TALEN. Ele continuou dizendo que, quando eles usam essa tecnologia, eles pretendem produzir uma quebra da dupla parte, da dupla camada do DNA humano, em um lugar preciso. Eles especificam qual que é o lugar em que aquilo vai acontecer. Mas o que pode acontecer é que a Cas9 pode, às vezes, induzir um corte em outros lugares do genoma. E isso é o que ele chamou de limitação off-target, limitação fora de alvo, se eu não estiver enganado, fazendo uma tradução bem literal. Então, a presença dessas mutações ou limitações fora de alvo é o principal problema que atrasa a aplicar essa tecnologia para as correções diretas in vitro. Continuando aqui, ele fala sobre o uso da tecnologia CRISPR com Cas9, que pode não ser apenas para induzir cortes no DNA, mas também pode ser usada para induzir a expressão de um gene, usando o Cas9 com a VP64, por exemplo, ou fundindo o Cas9 com o KRAB, KRAB, para reprimir a expressão de um gene, dependendo do tipo de doença que está sendo analisada. Então, no grupo de pesquisa dele, eles estão estudando o uso dessa tecnologia para desenvolver terapias de várias doenças. Ele citou a ataxia de Friedreich, a distrofia muscular de Duchenne, a doença de Alzheimer, e ele apresentou um pouco sobre o uso nesses tipos de doenças. No caso da distrofia muscular de Duchenne, 70% dos casos dessa distrofia, eles são devidos a ter exons deletados, e o número total de nucleotídeos codificados que estão deletados geralmente não é um múltiplo de 3, o que causa um conflito na cadeia, como ele mostrou nas figuras. Assim, é possível corrigir esse salto fazendo a remoção do exon 51 usando o RNA mensageiro. Isso foi feito com oligonucleotídeos. Este exon 51 também pode ser eliminado induzindo cortes nas suas bordas para remover completamente ele. Então, a remoção de um ou vários exons podem restaurar o quadro de leitura e converter um paciente da distrofia muscular de Duchenne em um paciente de Becker. Portanto, a melhora de uma distrofia muscular de Duchenne pode não ser significativa, dado que um paciente com síndrome de Becker pode estar preso a uma cadeira de rodas, foi um dos exemplos que ele citou. No grupo de pesquisa, em vez de tentar restaurar o quadro de leitura normal e excluir esses exons complexos, no grupo de pesquisa dele, ele tem o objetivo de produzir um exon híbrido que faria a ligação das proteínas do exon 50 ao exon 54. E ele mostrou também uma tabela que consegue predizer quais são os resultados esperados. Eles fizeram todo o processo e fizeram a indução em ratos de laboratório, e a sequência do exon híbrido 50-54 foi produzida em vivo, ou seja, dentro do músculo dos ratos, foi exatamente igual ao que era previsto. Isso representa um resultado muito positivo para o tipo de pesquisa. Finalmente, ele também explicou o uso da pesquisa dele, dessa tecnologia, para o desenvolvimento de pesquisas em relação ao Alzheimer, e o principal problema relacionado à abordagem de corte, como ele vinha falando do uso dessa tecnologia que veio através das bactérias, o principal problema com essa abordagem é que, embora desejamos modificar a citosina na fita, existem outros nucleotídeos de citosina muito próximos que vão acabar sendo modificados também. E isso conseguiu ser vencido através da tecnologia de edição Prime com a Cas9 Nikkase, que foi apresentada por um grupo de pesquisa, a Anzalone e os seus colaboradores, que apresentaram isso. De uma forma geral, ele conclui dizendo que as tecnologias derivadas do CRISPR com o Cas9 podem ser usadas para tratar muitas doenças hereditárias. Inclusive, eles estão entrando em trabalhos com a fibrose cística e a doença muscular congênita, entre outras doenças. E o principal problema permanece na entrega em vivo. Qual que vai ser o desenvolvimento disso em seres vivos? Então, eu agradeço pela paciência de vocês para essa retomada geral dos comentários e espero que eu tenha ajudado em alguma coisa.